Hello again. It's me, Steve Eath here, back for a second time today to make another short video because I wanted to share with you another aspect of the breast cancer cell line knowledge base that I haven't really shared with anyone before, but it's an aspect of the knowledge base that has basically been sort of the foundation for everything we've done up to now for a number of years now. As I've talked about in many of these videos, what we tried to build are several data mining tools that allow you, with a click of a mouse button, to figure out basically what's going on for any cell line with respect to the oncogenes that are activated, the genes that are expressed, gene essentiality, how that maps to KEGG pathways, all of these really interesting and very easy to use tools. All of these make use of a database that we built a number of years ago and have continued to build and are continuing to expand. And so I wanted to actually show you some things about the database because I wanted to give you a sense of just how powerful this kind of a database is, and to give you an opportunity to join me in generating novel queries of the database as well. So what I'm going to do now is share my screen again, and I'm going to show you basically just give you a sense of what this database looks like and how it works. Okay, so let's share the screen. Okay, so what you're looking at here now is something called MySQL or MySQL Workbench. SQL stands for Structured Query Language. And so one of the things that I've done over the last few months, partially as a result of working from home as a result of the pandemic, is to actually teach myself structured query language. And this has now given me a whole new opportunity to work with the database that I, along with some computer engineers, have built. And so I want to just show you this because it's very cool, and you may yourself want to query this database in ways that I have. So let me just give you some examples of that and just show you how cool and powerful this is. So up here is what's called the schema for this, what we call the Colt1 database. It's not important why we called it Colt1, but so each of these arrowheads point to a part of this relational database, and within each of these there are a number of, it's a huge table, and each table has a number of columns. And, and some of these tables might have, in this case, maybe 10 columns, but probably 15,000 rows. So the data for all the cell lines are present with regard to DNA amplification, cell line ID, the Achilles score, and all of that. So these are enormous tables that are present for each of these parts of the database. And this is, again, what's referred to as a relational database. So it allows us to write queries in ways that really allow us to ask some really very interesting questions with respect to this database. So if you just watch the previous video, I talked to you about the generation of tier 1, tier 2, and tier 3 drug sensitivities. And so the way I generated those for each cell line is using a query that's shown here in this window, and this is the query. And in this case, all I had to do to generate the tier 1 drug sensitivity is to enter the cell line ID for the cell line I was interested in. So in this case, the cell line ID number 40 it corresponds to that AU565 cell line. And so when I enter the number 40 and I click on this little lightning bolt, boom, this table gets generated showing the tier 1 drug sensitivities for this cell line AU565. Similarly, if I want to know the tier 2 drug sensitivities for cell line number 40, I just enter that ID, click, and the tier 2 drug sensitivities are returned instantly in milliseconds. And finally, for the tier 3 drug sensitivities. And here I indicated here cell line number 41, which is that 4475 cell line, the one with the BRAF mutation with the very interesting profile of tier 3 drug sensitivities. And so you basically just add, enter that, run the program, run the query essentially, and this table is generated. And and this is that PD drug that I talked about in that last video with their incredible drug sensitivities. And again, these are exactly the tables that are now on the website. And of course, once the table is generated, all it has to do is click on this little export button, and you can export this into a .csv file, make it into an Excel file, and save it and do additional analysis if you want. So, this is just an example how once you write these queries, running them could not be easier. You simply just type in the cell line ID, and that can be found in one of these other tables here. So that's not hard to find either. And you click on that button, and then you generate the table, and it's just that fast. So that's how I generated those tier 1, tier 2, and tier 3 drug sensitivities for the cell lines. We've got all kinds of other interesting queries that we've written as well that they really aren't on the website yet, but I wanted to just make you aware. So here's a really interesting query that we wrote that basically is aimed at looking at the association between any gene and its targeted drug in any cell line, and to be able to see that very, very quickly. So in this case, this gene ID 2064 is the gene ID for the ERB2 oncogene. And this gene, or the drug ID now, number 40, is the drug lapatinib. So if we wanted to know the association between ERB2 and lapatinib sensitivity, you just need to enter these IDs here and click on the lightning bolt again. And now you get very quickly this table. So this shows now for the gene ERB2. This is now rank ordering the cell lines with sensitivity to lapatinib by their Z-scores. And you can now see that in association with the amplification or the overexpression status, as well as the status of the screen, the data from the CRISPR or the RNAi screen. And so you can, again, find out very, very quickly the relationship between the presence of the amplification and the Z-score for the drug, and that allows you to identify some cell lines that have an ERB2 amplification but are not sensitive to lapatinib, and vice versa. And so again, all you have to do now is you can do this for any gene drug combination. So for example, we can look at the gene ID for PIK3CA, which is 5290, and the drug ID for alpelisib, which is 319. And you just sort of enter that in, and that's all you have to do to run the query. Click on the lightning bolt. Now you can see here's the PIK3CA. Here's the list of cell lines. Here are all the cell lines that have different PIK3CA mutations. And now you can see this decreasing sensitivity of cell lines that are with or without a PIK3CA mutation. So you're not too surprised to find that the cells that are most sensitive to the drug all have PIK3CA mutations. That's exactly what you would predict and expect. But now you can also find that there are some cell lines that do have the mutations but aren't particularly sensitive to the drug, some that have the mutation that are really not sensitive to the drug at all, and so forth. So you can really now see there very, very easily and very, very quickly the relationship between the presence of the mutation and the sensitivity to the targeted drug. And you can do this for any gene drug combination that you can think of that are in the database. Again, you find these gene IDs in this in one of these gene tables, the drug IDs in this table called dev drug, and then you just type them in, and it's very simple. And again, so we have some others. So for example, if you want to know the amplification status of any gene, you just type in again the gene ID here, and you click on the button, and it'll tell you, rank order these by DNA amplification, and then show all of the other data as well. Similarly, for point mutations, this is the PIK3CA again. You can just the query is written, and now you can look at all the cell lines with PIK3CA mutations, and you get the data for all the cell lines with and without PIK3CA mutations. I know we've got a lot of screen hit data in here that we use for a lot of our analyses. Sometimes you may just want to look at all the screen hit data though, and And this is the one, this is a query that allows you to look at just the rank ordered hits for, in this case, again, cell line ID 40, which is the EU565. If you want to just look at that data very, very quickly, I mean, you can't do this on DebtMap portal. It's very challenging. But in our database, you can just enter the cell line ID, and here in descending order, with the most essential genes to the least essential genes for the entire genome, there's 15,000 rows here. You can, and it's all sort of displayed by rank order, and you can look at this in this case for, this is the CRISPR screen. You can do the same thing for the RNAi screen, and you can compare which genes are most essential in both screens, and you can sort of figure that out by running these queries. And then of course we have oncogene signature queries and others as well. So I just wanted to sort of point this out to you, that these are all queries that I wrote, or I wrote in collaboration with some friends and colleagues who are into computer science. And because I wanted to know the answer to these questions, and I wanted to be able to extract this information from the database. And so I thought it would be interesting to just sort of show this all to you, because it illustrates the incredible power of the database. And I just sort of want to put it out there to folks who might have some other questions that you are interested in posing to the database, some other kinds of queries. And if you know how to write MySQL queries, send them to me, and we'll put them on the workbench, and we'll run them, and I'll send you the results. If you don't know how to write MySQL queries, basically just communicate with me and let me know what it is you want, what question you want to ask, or questions you want to ask of the database, and we'll write the queries and run it and send you the data that way. So this is a whole other aspect of the knowledge base. Again, as I indicated before, all of the data mining tools that we have are basically our Shiny apps that run on that database. So the database is really the foundation for the whole knowledge base. And I think we're just still learning how to maximize the power of this resource. And I guess I will say that one of the goals going forward now is to expand the database to other cancer cell lines. I'm working on ovarian cancer cell lines now, and ultimately we hope to have this expanded to many, many more cancer cell lines, so that we can do these kinds of queries for hundreds of different cancer cell lines. So I hope you find that a little bit interesting. It's a little bit different than the actual web-based knowledge base, but again, I wanted you just to have a sense of how all of those data mining tools are working, and again, give you the opportunity, if you have an interest in running queries to this database, we can work with you to do that, and like I said, either run queries that you write or help you to write queries based on things you're interested in. Okay, so that's it for today. Only 2 videos today. You're not going to hear from me again for a while. I hope you find them interesting and useful, and we'll talk again soon. Bye for now.